Welcome everyone. Good morning, first of all. Early morning for some of you. But we're here with Dr. Denise Furness, all the way from Sunshine Coast in Australia. Hopefully, once COVID calms down, we can take a visit there. But we'll be talking about optimizing fertility by addressing methylation and detoxification today. And if you've tuned in earlier and you tuned in a bit into our conversation, you'll just know that there's so much that we can do in terms of optimizing our fertility, in terms of even just our whole pregnancy journey. And Dr. Furness will be walking through some of these issues together with us today. So Dr. Furness is a PhD. She is also a functional geneticist and a nutritionist. She's a pioneer in the field of nutrigenomics, hence why we're talking about methylation today, and personalized health with over 15 years, probably more now.
Close to 20 now. We're getting old, close to 20 now.
Wow, okay, I'll have to update this.
Yeah.
Well, she began her research as a scientist focusing on folate, metabolism, epigenetics, in relation to pregnancy health. So she really is the person to be talking about this with us today. And over the years, her research expanded to covering vitamin D, immune markers, oxidative stress, and she also has many published works in peer-reviewed journals. She's won numerous awards for her research and conference presentations. And in 2012, so nearly 20, she founded Your Genes and Nutrition and began applying her knowledge in private practice. And she's also specializing in women's health, fertility, autoimmune disease. And I believe she also has a Facebook group where she does some lives. And so you can check her out on Facebook as well.
You can come along.
Yeah. So now I'll pass the time to Denise, the other Denise.
Thank you so much, the other Denise from Jara Health. I am so excited to be here. This is a topic that I love sharing because I do really know it in depth. I started in this world back in 2003 and actually talking about awards. So you can't see it. Just to have a little show off. Maybe at the end we'll see if I talk over time, then, but there is one up there. It's sort of talking about awards. I mean, I do so many different things now, not just fertility. I work with, after having my own autoimmune condition, I work with thyroid health, healthy aging, even around sort of Alzheimer's, dementia. But this is really where I started. And actually, there is an award up there from the Society of Maternal Fetal Medicine in the US. I won, to do with my research back then. So been doing this for a really long time. And back when I started, methylation, to my knowledge, wasn't a huge area, but particularly in functional medicine over the last 10 years, methylation has just exploded and a lot of people are confused. I did come to Hong Kong a few years ago when you could still travel, and we did a bit of a talk on methylation. So some of you listening might have met me in person. If you did, hello, and I will be coming back when I'm allowed to travel. But yeah, if you do have more in-depth questions, please let me know in the Q&A. I am going to talk about methylation, but not go really deep here. Really just try to help you understand what it is and what we can do about it as practitioners to help our patients, particularly in relation to fertility. So let's get started. Before I start, I would like to disclose I'm an advisor for Pure Encapsulations and that I have no other conflicts of interest to disclose. We are going to talk today about both male and female fertility. And if you did catch the beginning of Denise and I having a chat, I didn't realize you could hear me. So that's us just having a chat when I thought we were offline. It is really important to touch on both male and female fertility because so many women carry this burden on their own, and it is not a condition or an issue that is more common in women. It's the men and the women together that need to really work on their health. We'll talk about the foundations of methylation, common chemicals and toxins that can have a really big impact on hormonal health, on semen parameters, so the quality of the sperm, and fertility. This is really important. Some of the testing that I do, I think most of the tests I'm talking about, some of the more common pathology you'll have over there in Hong Kong. If you don't have some of these, let me know, or perhaps you even have other testing delving into some of the things I'll be discussing about. And then, of course, what everyone wants to know, what can we do from a therapeutic perspective, lifestyle-wise, which no doubt many of you working in functional medicine will know, but also key supplements that can really help boost fertility for our couples looking to optimize their chances of falling pregnant and having a healthy pregnancy and a healthy child. So fertility really is a big issue. And I have to say, when I started in the area, I was young. I was sort of, mid-20s, and I really didn't think that fertility was a big issue. I thought, once you, want to have a child, you'll just fall pregnant. My mum really didn't discuss any fertility issues with me. I do believe she had some. It is becoming a lot more common now for people to talk about it. But the reality is it's very common. It's common here in Australia. It's common all around the world, and particularly in Asia. So it's a global concern, and the use of IVF, or assisted reproductive technologies, including IVF, is rising around the world, including Australia and Asia. So let's start with male factor infertility. So 40 to 50%. And when I was working in the fertility clinic and in the public hospital here in Australia, we generally were sort of counselling our couples and would say that, 50% of the time, approximately, it is due to male factor infertility. So not only did we clinically observe that in the almost 10 years that I was working in obstetrics gynecology, but also there are numerous papers and publications from different parts of the world that also validate this. And the main reason that men do have issues with fertility is due to the quality and also, the quantity. So low sperm concentration, as well as poor motility, as well as abnormal morphology. So basically getting a semen analysis can then inform you of the quality and quantity of the semen and sperm. And what's really concerning, if you don't know this already, you may, particularly if you're working in fertility, is that sperm concentrations and semen quality have been decreasing over the past several decades. So there are studies showing that these semen parameters, the quality, when we are looking at motility, morphology, things like that. So how well do these sperms swim? Do they have structural abnormalities, morphological abnormalities? Do they have DNA damage? Is there oxidative stress? All of these things look like they are sort of getting worse and the quality is declining. And we do think, of course, we can't say this for sure, but it is hypothesized that this is due to the exposure to many of the chemicals and toxins in our environment, because our world is changing so rapidly. So when it comes to women, our fertility peaks in our 20s, but the reality is most of us do not want to get pregnant in our 20s. It declines in our 30s. We don't like hearing that, as well as our 40s. And there is a sharp decline after the age of 35. However, I am cautious with how I don't even really sort of say that to my fertility patients. The reality is people can get pregnant in their 40s. I had my babies, personally at 35 and 39, almost 40, thankfully, naturally. And it wasn't because I was, struggling for years to get pregnant. I focused on my career. I didn't meet my husband until I was in my 30s. And, from my knowledge, talking to Denise, but also just researching about rates of IVF and ART in Hong Kong, I think you are very similar in the sense that a lot of couples are leaving pregnancy much later in Hong Kong. You have a community that is very focused on career, and that's where a lot of their energy is going, and they're not thinking about having babies in their younger years. And so much so, if you did catch the beginning of the conversation, Denise and I were even just talking about trying to find a time that would suit you in Hong Kong. And please let Denise know to make sure that we can run these events at times that suit you. But 6:30 in the evening is when we've had some in the past. Now, to me, that's the end of the day. Work's done. But my understanding is in Hong Kong, people are working till, particularly the doctors, 7:30 at night. So they're the kind of shifts that probably need to happen, which are bigger than even fertility, because our lifestyle stress plays a big role in fertility as well. So talking about female fertility, let's get back to the specifics. The reasons that females are struggling with fertility is usually there's two things. It can be oocyte dependent, so meaning that there is an issue with the eggs, the quantity or the quality. And as we age, we know that the number of eggs decreases over time. You're born with the amount of eggs you have, and then as you age, you get less and less. And it's also thought that the quality declines as we age as well. But then there's oocyte independent. So meaning this has not actually got anything to do with the quality or quantity of the eggs that the female has. This could be something else, like they might have PCOS or Hashimoto's or some other medical, or they might have other things, structural abnormalities, issues with the fallopian tubes. I mean, obviously all those things are explored, and they can happen at any age as well. So I do want to emphasize too that fertility is not the only concern. When I started in this area, the first 4 to 5 years, I was working in a high-risk pregnancy clinic doing research in women who actually developed late gestation complications. So I didn't move into the infertility and recurrent miscarriage until later. So I was working with women that had experienced, late losses, so stillbirth, placental abruption, preterm birth, preeclampsia, or suffering intrauterine growth restriction. So the baby's not actually growing to that full potential. And that may be because there is an issue with the placenta, not getting the blood and the nutrients, or there's some issues with, either it could be a traumatic event, stress, or malnutrition as well can cause this. So you might have heard about the Barker hypothesis or fetal programming. So it's well established that adverse conditions, a little bit like what I just said with that intrauterine growth restriction, so lack of nutrients, or in some cases, in particularly in the Western world, it's not necessarily lack of energy. People are eating a really energy-dense, calorie-rich diet, but it might be lacking in a lot of micronutrients that are essential for optimal fertility. So the adverse condition could be that they're nutrient deprived, they have experienced some kind of stress or trauma, or exposed to certain chemicals and toxins. And this has been shown to increase risk, or it actually changes things in utero. It changes sort of what we call gene expression. We're touching on the epigenetics here. And this increases risk for adult onset diseases such as type 2 diabetes, hypertension, and obesity. So what we know is that conditions during pregnancy can actually change the way organs develop, change the way genes are expressed, and this has a lasting effect that can affect adult health. So David Barker back in the mid-90s was really the first to show this. He was looking at more sort of observational retrospective data, babies that were born during famines, and then showing that their risk of obesity and metabolic disorders was much higher. And since then, we have had tons of research validating this, a lot in animal models, some things you can and can't do in humans. But we do know now about these sort of epigenetic changes, particularly with changes in methylation patterns that we'll talk about. So the effects of this fetal programming, these things that are sort of turning off and on of genes, are likely to be wide-ranging and may impact on reproduction in the future generations. So it's hypothesized that babies that are born or that have been growth restricted, so intrauterine growth restricted, meaning the baby was, tracking along and then for some reason didn't reach its full growth potential. It's small, but not normally small. It's actually growth restricted. So these babies are thought to have a reduced number of follicles developing within the ovary. So these growth restricted babies are going to have, or more likely to have, issues with reproducing when they become adults. So we know that our environment plays a big role at influencing fertility as well as pregnancy health, and this includes our nutrient intake as well as exposures to harmful chemicals and toxins. And when we talk about our nutrient intake, we really have to hone in on our folate and B vitamins because these have a major impact on a process known as methylation. So let's get into methylation. What exactly is it? So a methyl group is literally a carbon and 3 hydrogens. And generally, when we talk about methylation, what we're talking about is a methyl group being added on to something. It's the process of a methyl group being attached to either a DNA, protein, or a fat, or some other molecule in the body. So it's the process of adding these methyl groups to a molecule in the body. And when these methyl groups are added, in the case of sort of gene expression, these methyl groups literally sit on the DNA and they block gene expression. So they literally turn genes off. In addition, it's not just about regulating our genes and turning them off and on. Methylation is important for both DNA and protein synthesis, and I'll show you that when we touch on the methylation pathway. It's important for energy production, detoxification, particularly in the breakdown of our catechol estrogens. So we use methyl groups via the enzyme COMT, catechol-methyltransferase. So these methyl groups are added to these estrogen metabolites to help make them less reactive. They could be metabolized, broken down, and excreted from the body. So methylation is essential for that regulation of our estrogen and metabolism of estrogen in the body, which then ties in with our hormone metabolism. And when it comes to hyper or hypo, and we're talking about sort of methylation throughout the body, we know that significant changes are linked with various health disorders such as cancers, as well as neurological and mental health disorders. Lots of research in relation to these sort of global methylation changes in the body. But focusing on fertility, we know that methylation is important for DNA replication. So when you think that you've got an egg that needs to be fertilized by the sperm, that then needs to replicate in from, a single cell into, hundreds, millions, thousands, millions and billions of cells to actually form that fetus. DNA methylation is essential for all those steps. The cell differentiation, what genes need to be turned on to make a certain tissue, to make a certain organ, as well as embryonic development, and again, that fetal programming. So this is what many of you as functional doctors and practitioners would call the methylation pathway. When I published it, this is back in 2008, we called it 1 carbon metabolism. It's the same thing because a methyl group's 1 carbon with 3 hydrogens. Or it can simply be called the folate and B vitamin metabolic pathway. It's all really the same thing. And I'm showing you this because for those of you who do delve into methylation, you're probably a bit familiar with the pathway, but also just to clearly show you the importance of folate and the B vitamins in making these methyl groups that then go and have all of these actions in the body, whether that's a methyl group adding to DNA or a methyl group breaking down a hormone, or a methyl group helping with detoxification. So methylation is actually one of the phase 2 detoxification pathways. Methyl groups can be added to things like arsenic to help excrete it from the body. So when we look at folate, dietary folate generally comes in the form of 5-methyl tetrahydrofolate. Now, folate comes in many different forms. It's not the only form, but predominantly. And also this is the most common form in our blood being transported through the body, the 5-methyl. So folate enters the pathway here. What it does is basically give a methyl. So dietary folate is 5-methyl tetrahydrofolate. So folate's providing those methyl groups for the body. This then donates a methyl group to homocysteine. Now, homocysteine is a non-essential amino acid. It's like an intermediate. It's not necessarily a bad thing unless it gets too high. That can then cause issues with sort of the endothelial cells, causing vascular issues. And that's also a really, really big problem when we're talking about pregnancy, because obviously the placenta is this vascular organ getting blood, nutrients, helping remove wastes from that growing fetus. And high homocysteine can cause damage to the blood vessels in the placenta, but we also know it's linked with cardiovascular disease and things as well. So we don't want our homocysteine getting too high. We want it sort of in a balanced range. And to do that, we have this methyl group added from 5-methyl tetrahydrofolate. Stick it on here. When the homocysteine and that methyl group is added, we actually get methionine. So methionine is an essential amino acid, and this is really important for cell proliferation and protein synthesis. Very important during pregnancy. And if methionine isn't being used for protein synthesis, it can also go on to form S-adenosyl methionine. Some of you might call this, or if you use SAMe in your clinic, you would call it SAMe. You don't generally call it S-adenosyl methionine. The supplement form is SAMe. So this is made in the body, and it's this S-adenosyl methionine that actually gives, donates, provides these methyl groups all over the body. So here you can see that this methyl group here, and then this can be used for sort of DNA methylation and gene expression, but not just gene expression. Those methyl groups can go off, as I said, to do many other things in the body. So there's a couple of steps here. I didn't mention them. If people have more questions about the genetics, I don't think a lot of the practitioners in Hong Kong are doing the genetics. I'm not focusing a lot on that, but super happy to ask any questions or answer any questions at the end if you want to ask more. But just know for that to happen, for that methyl group to be stuck onto the homocysteine, it does go through a few enzymes, and B12 is essential. If someone's low or deficient in B12, their homocysteine will go up. We kind of get a block here, and it does affect the pathway. So we need to make sure there's plenty of folate as well as plenty of B12. So what happens after this methyl group has been donated to homocysteine is this goes around, and we're left with this tetrahydrofolate. So the methyl group was provided here for the homocysteine. We've got this tetrahydrofolate here. So that continues around with the addition of B6 and another enzyme here, serine hydroxymethyltransferase, or SHMT. We then form this 5,10 methylene tetrahydrofolate. So this is another natural form of folate. And this can go in two directions. It can either convert uracil, which you find in your RNA, to thiamine, which is one of the 4 bases of DNA, and that is absolutely essential for DNA synthesis and repair. So there's 2 really major things going on here. We're providing methyl groups. We're also providing nucleotides for DNA synthesis or DNA repair. So really important. And you can see why, not just in pregnancy, but overall health, this is really important. Additionally, if this 5-methylene tetrahydrofolate, which by the way, if you have folinic acid comes in the pathway here, if you're familiar with that form of that supplement, this form of folate can then via MTHFR, which you might have heard about. It's a common SNPs in here that can be faster or slower that affects this conversion. But this form of folate via MTHFR can then be converted to the 5-methyl. And just a little note on folic acid. Folic acid in Hong Kong, I believe it is 400, or is it 500? 400 micrograms of folic acid is, if it's 500, just let me know, but definitely folic acid in Hong Kong is recommended for those wanting to fall pregnant. In Australia, it's the same. And folic acid, actually, when I was doing my research and we were giving supplements and testing things and pregnancy outcomes, homocysteine levels, folate, B12, the genes, etc., we did give folic acid. You know, that was more than 15 years ago, a long time ago now. And we did see benefits. The reality is there is lots of research showing this helps with neural tube defects. We saw it help with late gestation pregnancy complications as well. It can help with fertility. But the reality is this is a synthetic form. It's a synthetic form that has to go through a number of enzymes. So there's dihydrofolate reductase, DHFR. I wasn't going to talk about all this, going on a bit of a tangent, but yeah, there's a few enzymes here. It has to go through multiple steps to get to the tetrahydrofolate. So it's not that it's necessarily a bad thing, but what happens, and I might talk a little bit more about it at the end, is there's now sort of newer research coming out showing that folic acid can kind of saturate some of the receptors, and then can have some downstream effects. Also, for those that have genetic variations within MTHFR, MTHFD1, in all honesty, it is better giving more of these natural forms. Also, we have a lot of people that just have mitochondrial issues. They're not making enough ATP. They're struggling with their energy. Every single enzyme reaction does require ATP as well. And as I said, we can get this saturation of dihydrofolate reductase with the folic acid. It kind of blocks it up a little, and then things can slow down. So it's not necessarily that. I've heard some pretty striking and worrying things about folic acid, people saying it's really bad and does all these things. The reality is we don't have the evidence to suggest that it is. I mean, I've even heard people say it's toxic. That's not necessarily true. But the reality is, I think we are starting to see enough emerging evidence that some of these more natural forms could be better. And from a genetic standpoint, when I see lots of genetic variations within enzymes in the pathway, I want to provide a supplement, because we know that, for fertility, supplementing with our folate and Bs can be really helpful. There's no doubt about that. Tons of research. I want to be giving them something that is more easily absorbed, easier used by the body. Hopefully that makes sense. Please ask me any questions you have at the end. I am very, very happy and comfortable to talk about all of that in depth. All right, so as I mentioned, there are lots of studies. I think most of you be well aware that folate, B vitamins, and homocysteine, they do influence fertility and pregnancy complications, and pregnancy outcomes as well. So not only am I one of the ones that have done research here, there has been research all over the world showing this. It's well known that folate is required to reduce the risk of developmental defects, including neural tube defects. And as I said, most of the research at this stage really is with folic acid. And it is clear that it can help prevent things like neural tube defects, as well as other things like cleft palate and other developmental issues that can occur. And it's no surprise that the folate and B vitamins, when you think about that biochemical pathway I showed you doing so many things, helping provide amino acids, nucleotides, methyl groups, a gene expression, that there are multiple mechanisms, all those things I just mentioned, the DNA and cellular, that are supporting DNA and cellular replication, reducing homocysteine, which in itself can be inflammatory, can cause vascular damage, and supporting methylation, which is really important for certain genes that need to be turned off and on during those developmental phases. This is a study that was actually first published online in 2018, but it was actually published in hard copy. We had it published in 2020. So this was looking at MTHFR in association with fetal loss. So this was quite a large study that I was involved in, and one of the most recent papers that I've published in relation to 2020, because a lot of my stuff, as I said, was much earlier. But I want to make a point of saying that MTHFR, and some of you that works with methylation fertility, would be aware of this SNP. It's common. It definitely influences those folate levels. There are a number of studies linking it with fertility issues, but it's not the MTHFR itself. It's not the gene that's causing those fertility issues. It's the association with folate levels and homocysteine. So in this particular case, we also saw high homocysteine, so hyperhomocysteinemia in these patients with the MTHFR. And interestingly, we didn't see it in just the mums alone, and that's no surprise. In our previous studies, we actually didn't even see a link with MTHFR, even though lots of others had. And that's because we're actually also giving them folate, B12, B6. It compensates for these genes. But in this case, we actually looked at the maternal and paternal. We brought in the dad's genotype as well. And that combination of the mother's and the father's genotype, we saw there was an association again, but these mums also had the higher homocysteine as well. But it's not just MTHFR. I want to make that point if you are one of the ones doing genetics. I don't generally look at MTHFR on its own. There is those other genes. I didn't go through them in detail in the pathway, but they all work together. Those that affect that B12 availability for the homocysteine conversion. MTHFD1 is a really important one as well that's been linked with fertility. And I saw an association with this in relation to growth restriction as well. And no surprise there, because folate actually helps with obviously the growth of the baby, and that MTHFD1, similar to MTHFR, impacts that 5,10 methylene, one of those forms of folate. So let's go back to our methylation and gene expression. So there are other epigenetic markers, not just methylation. So there's acetylation, there's also microRNAs that can influence gene expression, but without a doubt, what we know the most about is methylation. And many studies have shown that methylation abnormalities, particularly within sperm cells, are linked with infertility. Now, it's not that methylation abnormalities aren't an issue within the oocyte or the egg of females. It's just that a lot of studies focus on sperm cells because these are much easier to work with, in the sense that we can collect these samples, not that men often like giving them, but from a research perspective, collecting semen samples is easier and less invasive than going in and collecting eggs. So lots of studies looking at sperm. We know that methylation, so those methyl groups sitting on the DNA, is highly dynamic during embryogenesis. So basically what happens when the sperm and egg come together and you have that fertilized egg, there's actually like a process of demethylation. All the methyl groups are gone. So the father comes along with his sperm cell that is methylated, depending on what he's inherited and his environmental influences. And then the mum comes along with her egg that has a certain methylation pattern as well. The sperm and egg come together, those methylation patterns are totally wiped. And then what happens during implantation, before sort of that blastocyst stage and before the embryo or the egg or the fertilized egg starts to replicate, the methylation pattern is then laid back down, and it's not random. It is dependent on the mothers and the fathers, but it's all put back. And there's really a very specific window here where the mother's nutrition is really important, because you need to have adequate folate and B vitamins to make all those methyl groups that you're going to be laying down on every single cell that is differentiated during that embryogenesis phase, or the development of the baby. We also know that the environment can have varying effects on DNA methylation depending on when the exposure occurs. So, for example, if you're exposed to a chemical or toxin during pregnancy, that could get in and actually have long-term effects on those methylation patterns that again could be handed on over the generations. So it occurs in utero. It's a lasting effect. However, if you're an adult and say you're smoking cigarettes and that changes the methylation patterns in your lung cells, that doesn't get passed on. It's not in the gametes, it's not in your sperm cell. But things that happen in utero, these can actually get passed on for generations. So speaking of that, this is a pretty landmark study. This came out in 2003. About the time I started my research, it was really concerning and really put quite a few of us, our heads in a spin trying to understand the impact that folate and Bs and our nutrient intake has on sort of epigenetics. So we did have obviously studies from knowing that people that had been through famines, there were likely changes in gene expression that were affecting obesity and metabolic health. But this clearly showed, this study by Waterland and Jertel, that changing the diet of mice that are genetically identical. So these mice are genetically the same. They are bred in the same conditions. The only difference was that their mothers were fed a different diet. So in the case of this particular mouse, the agouti gene was methylated, and it's actually meant to be methylated. It was methylated, the coat color was brown, and they had a normal metabolic profile. However, in this case, where the mother had a different diet, differences in the folate and the B vitamins and the methyl donors, including choline and betaine, their methylation pattern was different. And what happened is this gene was expressed. the coat color was different, the metabolic profile is different, and these mice were obese and prone to other sort of metabolic disorders. I believe it was, I think, sort of, a mouse kind of model of, diabetes, cardiovascular risk factors as well. So the only difference was the mother's diet. So let's talk about chemicals and toxins. So in the last 100 years, there has really been a rapid expansion in the chemical industry. These chemicals have affected, the things that we put on our skin, moisturizers, makeups, shampoo, the food we eat, the water we drink, and, a lot of these chemicals, particularly if you are in an industrial area or near a city, affect the air we breathe. So many of these chemicals have been shown to be harmful. And some of these have not been properly tested. And what hasn't been tested, even for those that are thought to be safe in very low levels, it's often the combination of chemicals coming together. I mean, how do we test for that? This kind of chemical soup that we're exposed to that possibly are causing reactions, themselves. And studies show that various chemicals impact fertility, pregnancy outcomes, as well as developmental defects. So when we think about detoxification, as practitioners too, we really want to support the liver. That's where a lot of it happens. It's not only in the liver. Obviously, gut health is important, kidneys, other things, but the liver is really taking a lot of the load with processing, metabolizing, and sort of helping eliminate chemicals and toxins from the body. So it happens in sort of these two phases, or really three phases when you think about elimination, but we've got phase 1, where there are a number of different reactions that occur. This includes our cytochrome P450 enzymes, known as the CYP enzymes, CYP. These are sort of involved in that first step of detoxification. And we know that many nutrients, and really I would say here folate rather than folic acid, many nutrients are needed to support this phase 1. So once the chemicals or toxins, it's not just exogenous chemicals and toxins outside the body, it's also endogenous chemicals too. Hormones and things like that need to be metabolized. After phase 1, we get these intermediate metabolites. And these are potentially, in some cases, even when we think about estrogens or caffeine, alcohol, things like that, when they go through phase 1, even benzenes, some of, lots of chemicals and toxins, as I said, whether they're from the outside environment or inside the body, they actually become more reactive. This intermediate metabolite is often a more reactive compound. So what we really need is the phase 2 working really well. And that's where a lot of people start to struggle. And this includes pathways such as sulfation, glucuronidation, glutathione conjugation. I'm sure many of you know glutathione being the master antioxidant, also really a big player in detoxification. So helping bind to these, and all of these, when we think about conjugation, these are, whether it's acetylation, amino acids are important, or methylation, it's the methyl group or the amino acid or the glutathione molecule literally conjugating, binding to that intermediate metabolite, making it more water soluble, less reactive, so that it can be eliminated and then passed through, whether it be through the bowels, urine, or even sweating, which doesn't say there, but I definitely talk to people when we're thinking about detox, if they can get into an infrared sauna or something like that, or getting out and about and moving their body and sweating and getting these chemicals and toxins out. So we want this to be working well, and we definitely want to be thinking about our nutrients. And again, I'm not delving into the genetics, but know that some people are a bit more sensitive. They might have deletions in some of the glutathione transferase enzymes that actually attach the glutathione to that reactive compound. If they've got MTHFR and other genetic variations in the methylation pathway, this could mean the pathway is likely to be a bit more sluggish. We want to support that, and we'll make sure at a minimum they have adequate nutrients to keep that pathway going, and making sure there are what we call cofactors. I didn't really explain that, but MTHFR, for example, the cofactor is vitamin B2, riboflavin, and actually studies have shown, not a lot of studies, it's more newer research, but we are getting some really good evidence that, increasing B2 increases that MTHFR activity. So they need to have enzymes So our genes, just stepping back, you've got our genes, our DNA, they're the instructions. They've got to make a protein that does the work. And for example, one of those proteins could be an enzyme. And enzymes generally require cofactors, which could be a zinc or a magnesium or a B vitamin to work efficiently. So when we do pick up some of these genetic variations, we can compensate, or at least try to compensate, with some of these nutrients and cofactors to boost that enzyme activity or help the protein work more efficiently. So when we think about exposures to chemicals and toxins, sperm cells are particularly susceptible to reactive oxygen species. So pesticides and environmental pollutants have been shown to reduce sperm count and semen quality. And there is a significant reduction in fertility in males with high concentrations of lead, other heavy metals too. This is just one example. But yes, other chemicals, benzenes, phthalates, things like that. And, phthalates, it's almost impossible not to have high phthalates because these are predominantly found in plastics. And, we live in a world where plastic is everywhere, but also things like fragrances and perfumes and things like that. And these are what we call endocrine disruptors as well. So influencing both female and males' hormonal health, which is really important for fertility. So sperm DNA damage is associated with infertility and miscarriage in both natural conception as well as assisted reproductive trials. And sperm DNA damage basically comes from exposure to chemicals and toxins that actually cause oxidative stress. So these reactive compounds, and they can actually do damage to the cell membrane, to the lipids, but also the DNA as well. So we often talk about oxidative stress with semen parameters and affecting male fertility, but there is no doubt that high oxidative stress, which again comes from things like, being exposed to drugs, cigarettes, alcohol, chemicals from the environment, but also if someone, doesn't have enough of those key nutrients, those minerals, the B vitamins, antioxidants. So antioxidants compensate for oxidative stress. And by the way, with oxidative stress, I actually cut out a lot of slides in the interest of time. I have given this talk before, but it was initially a much longer one. So I've taken out some of the explaining what oxidative stress is, and many of you probably do understand, but just know that it's not about having no oxidative stress. A little bit is normal and actually essential to turn on some things in the body, but it's all about the balance. So we want to have, antioxidants to help with those pro-oxidants that cause oxidative stress. So pro-oxidants being cigarettes, alcohol, exposure, even endogenous chemicals, hormones, neurotransmitters, etc., going through that phase 1 process in the liver. Also know that people that are obese, with particularly more adipose tissue around the middle, tend to be more inflammatory and have higher oxidative stress as well. And oxidative stress has been linked with conditions such as PCOS, endometriosis, as well as pregnancy complications and unexplained infertility. So an issue for men and women. And this is a bit of an alarming slide. I mean, it's not here to put fear in you. I think as practitioners, you probably know, but, BPA, which has been removed from plastics, this was back in 2012. But even when I went to New York in February in 2020, just before COVID, the last international event I went to, or the July, I came home and went straight to New Zealand, but then was only home 2 days later. But I was in New York at a conference in February 2020, and actually they were presenting data about the levels of BPA among Americans, more recent, still showing that despite us taking it out of the plastic, it's still in our environment, it's still in the water, we're still exposed to these things. So BPA is a hypomethylator. So BPA actually removes methyl groups and is associated with hypomethylation. The reason it's been taken away from plastics is it has been shown to affect infertility, developmental defects, and I don't think it's for sure, but some indication that maybe there are some links with cancers as well. So phthalates that are really common in the environment, flame retardants, which I'm not sure in Hong Kong, but here in Australia, they love to put this stuff in all the kids' pajamas. I don't buy my kids' pajamas often. They might just get like a t-shirt, some shorts, but yeah, flame retardants, really high levels in kids' pajamas, in mattresses and things like that. A lot of these chemicals can play havoc in the body, and they actually get transported, through the placenta into the baby. And this particular study, or this that was presented back in 2012, was showing that they looked at umbilical cord blood. There was 232 toxic chemicals. And actually my supervisor, Michael Fennick, back when I was doing my PhD in the early 2000s, we looked at DNA damage in the mother's blood in association with sort of late gestation complications. And he has been measuring DNA damage in cord blood over time. That's really a lot of what we did in that nutrigenomics epigenetics lab. We didn't just look at diet and genes, we also looked at DNA damage. And it does seem that DNA damage in the cord blood is actually increasing over time as well. So not only have we got our sperm, semen parameters reducing, exposed to more chemicals and toxins, babies tend to be born with more DNA damage. And DNA damage is something that does accumulate over our lifetime. But the less damage, the better when you are thinking about reducing risk of chronic disease and living a healthy life. Again, the same mouse, genetically identical, so the same mice being used, but a different study. In this particular study, they had mice that were exposed to BPA. So again, they're genetically identical, but their mothers during pregnancy were exposed to BPA, as well as having a slightly different diet. So the one I showed you before, or the only difference was diet. This one, we're seeing an exposure to BPA with a normal mouse chow diet, or exposure to BPA, which, as I said, is a hypomethylator, in addition with supplemented methyl donors. And supplemented methyl donors include choline. So choline, like folate, provides methyl groups. They used folic acid in this case, betaine and B12. And what it showed was that even though these mothers were exposed to BPA, which caused hypomethylation, and then this was obviously influenced the methylation patterns of their offspring, and they had the different coat color, obesity, and their different metabolic profiles. In this particular case, the sort of folate in bees protected against that BPA exposure. Now, mind you, I will say that I haven't read the study in quite a while, but the level of BPA that the mothers are exposed to was a higher amount, than we might be exposed to a tiny bit in water. So it is an animal model where they are giving them higher levels than we would hope any humans are exposed to. So common chemicals that I'm generally talking to my patients about include phthalates. So these are commonly found in plastics. Also think about plastic toys. If you do have children, a lot of those little rubber ducks and toys and things putting in the bath with hot water, no good. Solvents, household products, and approximately 2 million tons of phthalates are produced across the world each year. However, they're banned in Europe and restricted in the USA. Phthalates are an endocrine disruptor, and studies have shown that they do cause developmental defects, particularly within males. And again, this all comes down to dosage. I don't understand why they're not totally banned in Australia, though it's hard because we import so many things. And actually speaking to people in the US, it's, they want to ban it, but the reality is, it comes from particularly a lot of things coming even from China actually can have higher levels of phthalates, but in the US, they actually test the plastics and things coming through to make sure they have really, really low levels. But yeah, when it comes to developmental defects in males, again, this is sort of with higher levels, but we need to think about the accumulation. Even though a tiny bit might be thought to be safe, the reality is these things accumulate over time. If people are having problems with their liver, they're not detoxifying properly, they're not using their bowels, they're not sweating, they're not getting the right nutrients to help these things. They've got genetic predispositions that mean they're not as great at detoxifying, plus they're exposed to lots of other things. It's all of this coming together. So I'm not saying specifically one tiny amount of a chemical, it's that bigger picture that you would do as a functional medicine doctor. You're looking at that holistic approach and the whole person. Parabens, so these are the most common preservative in skincare. Parabens are endocrine disruptors, and they have been linked with cancers and skin aging. And what I find fascinating is that parabens, can be found in some of these really expensive face creams that are costing, hundreds of dollars. But there's studies showing that parabens actually affect skin aging. So they're not good for our skin. I have to say, though, in the years of learning about this and sourcing more natural things, not just for fertility, but for overall health, and me having an autoimmune condition and working with all my patients, it is becoming a lot more known that things like phthalates and parabens are no good, and it's becoming easier than ever to actually get. And it doesn't have to be expensive either, but more natural skin care, more natural personal care things, you know, whether it be shampoo, moisturizers, makeup, things like that, that don't have these harmful chemicals that are known to be endocrine disruptors. Heavy metals are often a concern as well, particularly mercury from fish. This can have issues on our nervous system, immune system, and there have been studies showing that particularly levels of mercury can affect developmental defects in utero and early life. Benzenes is another one that I talk to people about. So I do some testing where I do look at environmental pollutants, and benzenes is one that comes up a lot for people that live in cities. It's ranked one of the top 20 chemicals for production in the USA. And really it comes from, think about burnt substances like tobacco smoke, petrol and exhaust fumes, industrial emissions. But it can also come from indoor air, not just outdoor air. So it's not just if someone's living near a big freeway or a big highway, if you call it that in Hong Kong, but you know, those busy roads, but also like the motorways, but you know, indoor air can be affected by these things as well. So how do we test and find these things out? Well, if we sort of go back to our nutrients, we want to be looking at things like folate and B vitamins, homocysteine, which I've given an asterisk here, because if you are limited with the testing that you can do, homocysteine is probably the one that I would test for a methylation marker. You can look at S-adenylyl methionine and S-adenylyl homocysteine, that ratio that helps give you an idea of methylation. Doctors' data do have, there's probably a few of the functional medicine testing companies now, but Doctors' data have one that I occasionally use. I don't do a lot of it. You can look at genetics, lots of different companies doing that. We can talk about it at the end if you like. And then when it comes to methyl groups, when I was in the lab and had a lab, I did test actual methyl groups sitting on the DNA in the mother's blood as well as placental tissue. But I don't know of any good commercial test to be able to really look at that. So we're looking at other markers to get an idea of someone's methylation, you know, the methylation. So if the homocysteine is really high, there's probably some blockages or it's not moving well. There are on occasions where the homocysteine can be quite low as well. And that's an issue because you need homocysteine to make methionine, to make the methyl group. So we want it within a nice range. And I will give you those ranges. So if we want to look at folate, you can talk to people about their diet and nutrient intake. So doing a food frequency questionnaire or a food diary. You can look at red blood cell or serum folate blood levels. Just even looking at a full blood count. Obviously, if there are changes in that, that can suggest possibly anemia, which can be linked to folate, B vitamins or iron. If you do functional testing, you can look at some of the more natural forms like the derivatives, like 5-methyl tetrahydrofolate. That's not as common though, and can be expensive depending where you get the testing done. And another thing I like to do that some of you might do is organic acid testing. So I personally use Great Plains, and Genova is a really popular one over in the US as well. But with Great Plains, I get this uracil or thiamine marker. And if you think back to the pathway, you may or may not be with me right now, but if you visualize that methylation, that folate B vitamin metabolic pathway I showed you, I sort of said that folate or, you know, methyl groups are needed to convert uracil to thiamine. So folate donates a methyl group, sticks it onto uracil to make thiamine. So this is a bit of a marker of functional methylation. We can get that from urine metabolites. Or there is another marker we call FIGLU that can build up when someone is having methylation issues. So these are other markers you can look at as well. When it comes to B12, again, finding out about their nutrient intake. Are they vegan? Are they vegetarian? You can measure B12. In Australia, we also have HOLO, so just our normal pathology testing. We get not only the serum total B12, but HOLO transcobalamin, which is probably a better marker than the total. I mean, this is not a really robust marker at all. Full blood count, again, similar to folate, giving us an idea if there are some issues with maybe the Bs or iron. Methylmalonic acid, which I think is the best marker to look at B12. This builds up if someone's deficient in B12 or low. And then homocysteine too, a bit of an indirect marker. If someone's low in B12, this will build up. So when it comes to homocysteine, this is a sensitive functional biomarker of either folate or B12 deficiencies. We don't necessarily know whether it's the folate or the B12, but it's letting us know there's a problem in that pathway and it's building up. However, it's not a specific marker. Generally, most of the time, it builds up because of low folate or B12, but there can be other things. The person might have problems with renal insufficiency or certain drugs. And although there's no formal standardization program for homocysteine, they generally have good performance such as accuracy and precision. And I say that because things like serum B12 do not. They don't tend to be a robust marker, and there isn't good performance or accuracy between labs and between countries for the serum B12. Looking at the reference ranges, we in Australia, the reference range goes between, it's about 5, depending on the lab, 5 to 15 micromole. The Mayo Clinic says that under 13 is, or equal to 13 is best. Over 13 is considered abnormal, and generally less than 10 is desirable to reduce cardiovascular disease. And even back when I started learning about this in the early 2000s, there were studies showing that really anything over 9 does link with increased risk for cardiovascular disease. When we talk about optimal ranges for pregnancy, so this is sort of coming into the pregnancy or, in the early stages, homocysteine does drop by about 20 weeks gestation because, no surprise, it's being used up to make methionine for methylation for folate. So it does go down. You really don't want to be below 6 because it could get too low. But from my studies where we were looking at homocysteine levels in early pregnancy, to reduce risk of pregnancy complications like preeclampsia, growth restriction, preterm birth, really around this 6 to 9. Michael Fennick, who is my supervisor, who's really the guru of nutrigenomics, known all around the world, who looks at DNA damage as well. He says less than 7.5 homocysteine is associated with lower risks of DNA damage in the blood cells. And Dale Bredesen, who some of you may know if we're talking about Alzheimer's, really suggests, particularly for those that already have early cognitive decline and you want to reduce that cognitive decline and reduce risk of Alzheimer's, 5 to 7. So they're all pretty low levels, but just know anything under 5 is actually not good either because you do need homocysteine to help make the methionine. Also, I didn't mention it, but it goes down the transsulfuration pathway, helps make glutathione, taurine, and does other things as well. And if you do homocysteine and you're wanting to work out if someone's, low in folate or B12, work out why it's high, you can always do a methylmalonic acid to let you know if the B12 is the issue. I don't necessarily go that far. If you've got high homocysteine, I generally like to give a B complex with folate, B12, B6, B2. They all work together, so it doesn't necessarily matter. You want to be giving that B complex to get that methylation pathway working efficiently. When it comes to chemicals and toxins, you can also do questionnaires, finding out about how many products someone uses or if they're exposed to certain things, blood and urine testing. There's also tests here in Australia. I'm not sure if you've got them, looking at oxidative stress levels. And I also do like to look at genetic susceptibility, as I mentioned. This is just a paper to show the benefits that you can get from just even a questionnaire, even if you don't have the ability to do some of these more expensive tests. So this study showed that by actually asking people and doing a questionnaire about, self-reported personal care, so using lotion, so moisturizers and things like that, cosmetics, perfume, they showed that the people using the most of these personal care products did have the highest levels of parabens and phthalates. So even just asking some of these simple questionnaires in your clinic, and then finding out and getting your patients to turn around and read the back of the bottle, the labels, and get them to swap the products that they are using, because these things are endocrine disruptors. They're associated with increased oxidative stress, abnormal semen parameters, and therefore fertility as well. All right, so what can we do to improve fertility? So there's no doubt that reducing alcohol and drugs is one of the first things, because this not only depletes our levels of folate and B vitamins, we need these to sort of metabolize them through the liver, but also increases our risk of oxidative stress. Stress is a big one. We could do an entire talk on stress. It's an issue for so many people. Maybe lots of the people in Hong Kong too that are working really long hours. But yes, it does deplete our folate and B vitamins as well. So adrenaline, one of these kind of stress hormones, actually needs folate and Bs to be broken down. So talking to people about strategies to manage their stress. Sleep is obviously essential. Most adults benefit from 7 to 9 hours, going through those different sleep cycles so that they can, flush out the brain, helping with memory learning, but also turning on the immune system at night, producing growth factors, for healing, for repairing. A lot of that repair happens at night. If people want to get pregnant and they want to get well, sleeping is something that needs to be addressed as well. Physical activity as well. People need to move, but of course, when we're thinking about fertility, don't overdo it, because the reality is exercise too does cause a bit of oxidative stress, but a little bit is good. A little bit is definitely good. We're all made to move and not be sitting in front of computers all day, every day, which so many of us do. When we want to minimize our exposure to chemicals and toxins, and that's really one of the predominant things you need to be focusing on, filtered water, clean air. Perhaps some people that are living, right in the city need to consider an air filter. Other things, plants. I have lots of plants in my house. There are certain plants that actually clean the air, that actually remove benzenes, things like that. So thinking about some nice plants, they look pretty, but they'll also help clean the air. Diet wise, we're talking about a whole food diet with lots of, natural whole foods that'll have lots of antioxidants, a wide variety of vegetables, predominantly vegetables, a more so plant-based, not necessarily vegan or vegetarian, but a plant-based diet with lots of veggies is really good for reducing oxidative stress. In addition, fasting or calorie restriction has been shown to reduce oxidative stress. For men, they could trial a 16-8, so 16 hours of fasting, 8. For women, a lot of the research that shows this is beneficial is in men, particularly athletes. I think for women, 14-10 is generally something that's more achievable, more lasting. We are different. Our hormones are different. So 14-10 generally for women. We also don't want to be fasting. These are things you can be doing your preconception, losing weight, doing a bit of fasting. But once they're actually trying to get pregnant, we don't want to be sort of detoxing anymore. We don't want to be trying to sort of lose weight and things like that, because a lot of these chemicals can be released into the body, and we want to kind of try to get most of them out before the mother gets pregnant, so they're not getting pushed through that placenta and being stored in the baby. Minimizing intake of processed foods, and considering things like herbicides, pesticides, flavors, organic if they can, really important during that preconception and pregnancy phase. There are many foods that are rich in folate, green leafy vegetables, particularly legumes, things like that. B12, this is really our animal product. So if someone's vegetarian or vegan, you may need to think about higher doses of supplements with them. And really, when it comes to it, a good quality prenatal, there is no doubt that a good quality prenatal is the best. As I said, I know that in Hong Kong, it's the same in Australia, folic acid is suggested, and that really comes down to those early studies, those randomized controlled trials that showed that folic acid reduced risk of neural tube defects. But the fact is, I showed you the pathway, folate doesn't work on its own. And if you don't have the B12, the B6, the B2, things can get stuck. It's actually called the folate trap, where the pathway is kind of stuck because, without that B12, it can't do the next step. Without the B6, it doesn't do this. So thinking about something with all of those Bs, and then I haven't talked about it in this particular lecture, but if we're thinking about, optimal, fetal health and things like that, we want to have some iodine. We want to have, other nutrients that potentially can be low that are going to affect brain development, things like that. When it comes to fertility boosting, there is research around CoQ10, particularly for those older parents, quite a few in relation to improving semen parameters, but even some studies. I saw one recently, just published the end of last year, suggesting that those doing ART, a little bit of CoQ10 added to the media helped with the development of the embryos. So yeah, CoQ10 has got quite a bit of research in reducing oxidative stress, as well as helping with semen parameters. So that's definitely something I think about. There are different forms of CoQ10, straight CoQ10 or ubiquinol. If you've got more questions, we can talk about that at the end. Omega-3s, so this is tons of research here, and this is something that we did give back in the fertility clinic as well, along with our prenatal. Generally with the omega-3s, to get to sort of therapeutic doses, you are wanting to often have higher amounts. You don't want to be taking, 10 or 12 capsules. I know that Denise does have the omega-1, which has that higher dose of EPA and DHA. I think it's 600, is it 600 milligrams of EPA and about 400 milligrams of the DHA. So that could be, you could have a single, if your patient is having, omega-3s, I think you tend to eat more seafood over there in Asia, but remember the smaller fish, smaller things that aren't going to be building up mercury and things like that, you might only need one, otherwise 2 capsules. If they've got any if they have any inflammatory symptoms, like a bit of joint pain or things that you think inflammation's an issue, maybe you want to go with 2 capsules. But that's generally a good one, because the others that have a lower dose, I mean, they're fine, but you've got people taking quite a few capsules, and not all patients like that. Antioxidants are key too, compensating for that oxidative stress, those exposures to things. Yes, we can minimize exposure, that's number one, but the reality is we are exposed to lots of things. It's a particularly crucial time, preconception and pregnancy. So antioxidants too, that not just thinking about, your A or your E or things like that, but even things like zinc and selenium and stuff like that, that actually help work with the antioxidant enzymes in the body. And that's actually, there's studies too, that these are really important for reducing oxidative stress and DNA damage in sperm as well. So there is an antioxidant formula that I like, that again, I know Denise has in stock, that actually has some antioxidants along with the selenium and the zinc as well, which is really good for men that are showing issues with semen parameters. And of course, addressing any nutrient deficiencies. If someone's low in vitamin D, we need to give them vitamin D. If there are issues with iron, give them iron. But these are kind of my go-to, in the sense that, definitely a prenatal. And then for fertility boosting, without a doubt, these are my priorities. And again, quality is essential. If we're talking about fertility too, we don't want someone having supplements that's got nasty excipients or things that could actually potentially be reactive. So thinking about a high quality supplement, it's going to sound like I'm just plugging Pure here, but yes, I am, but it's not about pushing a product on you. It's just knowing that Pure Encapsulations does actually care about what's in their supplements. There's third-party testing. They send things back if they don't meet certain standards, things are tested. So you just want to make sure that you've got a supplement that is clean, that doesn't have other nasties, and that you can be sure of the amounts of the particular nutrient that you're getting. So really consider that in the fertility case, because we don't want to be putting in anything that's going to cause more oxidative stress or reactions in the body. When it comes to dose, sorry, I thought we were at the end a little bit more here. Again, we can talk about doses in question time. Generally, 1 milligram is my maximum for folate. But of course, if someone has had neural tube defects, we do go to that higher dose, the 5 milligram. But I really only do that for certain populations. Generally, not going over the 1 milligram. I like 500 to 600 micrograms is generally good. You need to have the B12, the 6, the B2, etc. I like a B complex rather than folate on its own. And when it comes to folic acid, I did mention this in the pathway. Studies show there's possible saturation affecting that enzyme, and then actually the whole pathway can start to slow down. But in saying that, the reality is when you trawl through the literature, the medical research, most of the evidence supports folic acid, but emerging evidence suggests it may not be the best form, particularly at high doses. And I think a lot of the issues with folic acid is that people have just taken way too much. And also a lot of our foods are fortified. We want to be thinking more about natural forms. And as I mentioned before, those that have particular genetic variations, I'm definitely thinking about, the natural forms like the 5-methyl tetrahydrofolate. That would be my preference because you're directly supporting that methylation pathway. Same with the B12. You can have methyl B12, hydroxycobalamin, adenosylcobalamin. If we're thinking about supporting methylation, that methylcobalamin. Just finally we do talk about preconception care, giving these supplements to help our couples get pregnant, but it's not just about getting pregnant. This is a study that, I was involved in. It wasn't a huge cohort, but we were measuring blood levels of, folate, B12, things like that, 20 weeks, so mid-gestation. And we found that those with the lowest levels of folate mid-pregnancy were more likely to develop small for gestational age as well as IUGR. So I do tell my women to supplement throughout pregnancy. It's not like as soon as you're pregnant, you don't need these nutrients. Actually, your nutrient requirements increase during pregnancy and breastfeeding. So don't forget to support your patients throughout pregnancy and throughout that breastfeeding stage as well, assuming they can breastfeed. All right, we are at the end, people. Thank you for staying with me. So in summary, our diet, nutrient intake, and lifestyle changes can really have a big impact on fertility through supporting methylation and reducing that toxic load. Timelines, we can talk a bit more about this at the end. Most of our patients come and want to get pregnant instantly. I do like to work with them for about 3 months. Think about those sperm cells. It takes about 90 days for that sperm to go through the process of developing, being ejaculated. So it's nice to sort of start with at least 3 months. And as I said at the start, IVF, assisted reproductive technologies, they're becoming more common. Infertility is an issue around the world. And by working in this area, providing comprehensive prenatal care, we really can help our patients not only improve fertility and getting pregnant, but remember, it's the lifelong health of that child, reducing risk of adult onset diseases, and of course, helping improve the health of the mums and dads. Thank you so much, everyone.
Thank you, Denise. That was great. And I think, even if, we currently don't have patients we're working with in terms of helping them through their fertility or pregnancy journey, this is really helpful just in general, like with the methylation, as we see that it's really important, not just for pregnancy, right? It's also for one of the slides that you have for, with chronic diseases. I'm one that can't process benzene. I think you helped me read one of my reports, and I don't process
the
benzene.
NQ01. So that actually ties into the CoQ10. I think you had a genetic variation NQ01, quinone reductase, that actually helps, with getting rid of the benzene. But also it's what helps convert CoQ10 into ubiquinol, the active form. So you're someone, if I was working with you and wanted to give you CoQ10, for example, because it's mitochondrial support. And we know too, when we're thinking about fertility, particularly aging couples, we do want to think about mitochondrial support as well. And that's sort of where the CoQ10 fits in. Plus it's an antioxidant. But the ubiquinol would be a great form for you. And if we are talking about giving CoQ10 as a supplement to our patients, I do tend to prefer the ubiquinol because we don't always have genetics, but the reality is that is more expensive. It is the active form. You're putting in something that can act as an antioxidant straight away or help with, metabolizing benzenes, things like that. So, and helping with supporting the mitochondria energy production. So I do like the ubiquinol. However, the CoQ10, there are a lot of studies, and when we're talking about fertility, a lot of the studies actually showing the benefits are with just the CoQ10, not necessarily that active ubiquinol. So there is quite a substantial amount of evidence just showing the straight CoQ10 is really beneficial as well.
What's the dosage, just out of curiosity for
Yeah,
myself?
so the dose is really wide in the sense that, it's probably somewhere between like 100. I actually just had someone come to me too, and he was on 600 milligrams. And a lot of supplements are around 100, 120, 150, but he was on 600. And some fertility specialists, so he's actually seeing a fertility specialist with his wife, and he was on 600. I would say generally about 300. That's probably a good way to go. The ubiquinol that I use is actually either the 250 milligrams, so they can just have one a day. Particularly with men, they might just want to have one. If you've got really compliant patients, and one great thing about fertility is your patients are generally quite compliant, quite motivated. It is also nice to kind of spread it out throughout the day. So maybe you want to give them, 100 milligrams in the morning and 100 milligrams so the CoQ10, you might be looking at, so let's just say there's 120 milligrams in the CoQ10, you might give them 120 in the morning, 120 in the afternoon. I do like to give a little bit more. So that'd be, 240. I do like to give a bit more of the CoQ10 rather than ubiquinol. But yeah, kind of, between that 200 and 300. And again, depending on which supplement you use, you might just give them a higher dose of, 250, or you can spread it out throughout the day. And if you are spreading it out, don't give it to our patients right before bed. There are some studies, remember, it is sort of interacting with mitochondria and energy too. Some studies suggesting it might even link with insomnia. So, and the same with the B vitamins too. The B vitamins are best in the morning, not at night. So there are great supplements at night, love some magnesium at night, some probiotics at night, but the timing of our supplements can actually, be important as well. So the CoQ10 earlier in the day, morning, early afternoon.
Okay, great. Just seeing if there's any questions popping out. Doesn't seem to be any. But I really liked your suggestion on the questionnaire, because actually in Hong Kong, I'm not sure for those listening whether you're aware, but Hong Kong had only, I'd say in the past 2 years, just started banning microplastics.
Great.
In our beauty products. And just recently received an email about banning partially hydrogenated oils. So we're getting there, but the reality is that we do live in, this is a globalized world where Hong Kong is a free port. I don't believe that they're testing everything that comes in, like probably Australia or the US. They are doing some batch testing, but I don't think that they're testing everything. And we are, neighbors with China.
I know.
Where a lot of,
yes.
the regulations are less stringent. But like, for example, the partially hydrogenated oils, that's not even going to come into effect until 2023. So these are just having like simple questionnaires, like even if we're not doing blood work, a simple questionnaire could definitely shed some more light.
Yeah, even just, doing a food diary. I mean, so many patients will come and say, I have a really good diet. And then you start looking and you're like, oh, really? we can tweak this here. Or they might tell you how many things, how many products they're putting on their skin. And, some people a lot. We live in a world where, think about the advertising, the magazines, and, use this. And I can't think of the numbers. I've given talks years ago where there was a study, a British study, and it was crazy. It's, over 100 and something chemicals that most women put on their skin each day. Now, that was an older study. I do think things are changing. And as I said, products, particularly here in Australia, maybe in Hong Kong too, like even just at our local supermarket now, the shampoo will say phthalate-free, sulfate-free, paraben-free, all these kind of things. So it is becoming really common and more well-known, which is great. However, there are some people that still just don't know. And there are some products that, it's becoming more well-known, but I wouldn't say it's not everyone. So yeah, and particularly when we're thinking about fertility, I mean, really anything, even if you're thinking about neurological health, hormonal health, menopause, a lot of these endocrine disruptors. Yes, they're going to affect fertility, but they can also make women really, really struggle through menopause. So, we want to be eliminating these things. We want the body to be working as efficiently as possible, and getting rid of some of these nasty things. So eliminating is definitely number one. Obviously, we can bring in some supplements. I mean, I mentioned the antioxidants. I like the antioxidant formula because it's got a couple of antioxidants and some minerals. But there are other things too that I love, like NAC, N-acetylcysteine, or glutathione. These are great. So if your patients do come to you, you do a questionnaire, or you find out that, even if not just what they're using, but talk to them about their symptoms that can be linked with a high chemical or toxic load. If they're getting headaches when they smell petrol fumes, or, headaches to perfumes, or they're getting a lot of rashes, or they've got some sensitivities to foods, they're noticing, hay fever, sensitivities, all that stuff, that's a really good indication their toxic load is building up. They're becoming really sensitive. Their immune system's, on high alert. There's lots of oxidative stress. So inflammation and oxidative stress basically tie in together. So if they've got all of these symptoms, you're like, this person's really sensitive. I think they've got, an issue with, a high level of chemicals and toxins. They tell you they're constipated, they're not getting the waste out of their body. You can then start to think, well, I need to really support the liver here. I mean, one, you want to get everything out first. So think about the constipation. But then bringing in things like, glutathione or NAC, or things that are really going to help with detox, maybe even binders, depending on what you think they're exposed to, if it's molds, charcoals, clays. Now we're going all sorts of things for you as practitioners here, but all the things you can do to support detox, but you do that before they start trying. So that was when I said about the 3 months. People don't like you saying, hey, I want to work with you for 6 months. So tell them 3 months, minimum a month. But if they are, quite sensitive and they're unwell and they've got a lot of health issues they need to address, you want to be doing as much of that as possible before they actually start trying. And I think when you explain it, and they do start to understand that they're often having a lot of other health issues as well, they can come to terms with that. But sadly, most of the time, I know a lot of the patients, not so much now, but years ago, when I moved from the public hospital to private, a lot of the patients I got, they'd already been through so many rounds of IVF and they're in their 40s and they just want to get pregnant straight away. But now I do tend to be getting younger people in, which is great. And I do have a bit more time and flexibility, or they're not in such an urgent rush. And that's really, really nice, because we want them to be in the best, health possible.
Yeah, that's really important. We do have a question in that. This will probably be our last question just because of time. I don't want to keep you. I know it's later there. But
thank you. And I did go over time. I'm sorry,
Yeah.
I went over time. I actually cut a lot out. I was like, I won't be too long. It was actually it was 2 talks combined to one, and then I cut a lot out. So sorry I went over, but I can see everyone stayed with me. Thank you.
But in terms of prioritizing testing, what are the top 3 tests that you would use? And for glutathione, is there an overdose that we would need to watch out for?
Yeah, so always thinking about, dosages, not going too high, interactions, even the CoQ10, even though I really like it, thinking is a person on blood thinners, things like that they could possibly, antidepressants, CoQ10 can interact with some of those things as well. Obviously the same thing with the omegas. When I start talking about higher doses, having a couple of capsules, or you might really think they've got a lot of inflammatory stuff going on, you want to give higher omegas, again, think about interaction with blood thinners. But yeah, the glutathione, what I do, so I personally, if someone is what I call sensitive, like what I just explained, they're reacting to smells and some foods and getting rashes and, headaches and signs that they're sort of got this toxic overload. Even though a lot of practitioners love glutathione, and I've had some practitioners tell me it's like the wonder drug, I actually start with NAC. So I like NAC. I find that less people kind of, because sensitive people are sensitive. You really actually, even though pure encapsulations have got high quality supplements, some people react to some of these just little things. It just, their body's quite out of balance. So I would be thinking about NAC, which is basically cysteine, and that is the precursor to make glutathione. So actually homocysteine, what I talked about, has to come down to form cystathionine and then cysteine. Our body can make glycine and glutamine, which are the two other amino acids to make glutathione. So cysteine is the sort of limiting factor. And studies do show that by giving NAC, it increases glutathione. So you're less likely to get that kind of reaction to glutathione, because I have had, even actually myself personally, when I was recovering from my autoimmune stuff, it's probably why I'm a bit more inclined to use the NAC too. I had glutathione, was told how great it was, and I actually just came out in so many pimples. Like I had a reaction with my skin. And I have had other people tell me that it makes them feel nauseous and things like, I mean, these are rare, by the way. I want everyone listening to know these are not common. Most people can tolerate glutathione. It's really, really good for the body. But you do have these highly toxic, chemically sensitive people. My suggestion would be to start with NAC. A dose of NACs somewhere between 500 milligrams up to a gram, but you might even want to start lower than that, and always with people that you are suspicious that, and again, if they're not using their bowels or they're not clearing their waste properly, really low doses, because otherwise they do tend to react. I don't know, Denise, do you tend to get a lot of, are your practitioners, or maybe someone can comment, are you seeing a lot of really sensitive people? I tend to get a lot of doctor referrals for patients that doctors don't know what to do with, like they're super sensitive. So I do see some of those people. And again, why I probably align with Pure is because I'm looking for supplements that I'm very confident with, because I do get these people. 15 years ago, 20 years ago in the public hospital, I never saw this. It was a very different population. But I'm now working with people with major food sensitivities, allergies, they're very autoimmune, and we really have to tread cautiously with these people.
Yeah. I mean, I take NAC probably more on a day-to-day basis. And then if I want to do a deeper detox or a seasonal kind of detox, then I kind of switch over to the glutathione for that period of time. Yeah.
And I was thinking
regarding just fertility testing quickly, what
yes, the testing, sorry. Yeah, so fertility testing, as in testing for the male and the female, without a doubt, semen analysis for the male, find out if his sperm cells are good. For the female, I guess all the fertility stuff that you would be doing, checking to if there is a structural abnormality. I don't really do a lot of AMH testing anymore. I used to when I was at the hospital. So AMH is that anti-Mullerian hormone. We call that kind of egg timer test. If it's low, it might suggest that, or it generally suggests the person's got low eggs or low quality eggs. But I don't do the AMH testing anymore because it just puts more, I guess, stress for someone to go, oh my God, the reality is my treatment isn't going to change. Whereas with the male, knowing that they are a big factor, then they can be really involved in getting onto the supplements, really changing their lifestyle. The women tend to not have any issues with, wanting to change diet, wanting to change lifestyle. When you say to a male, I want you to stop drinking this much coffee, I want you to stop drinking alcohol, I want you to stop smoking, I want you to take supplements, that can be a lot. And again, I'm generalizing. Sorry to the men that are out there that, maybe don't agree with this, but this is what I see. So I think having that for them to know that, the issue with the male, but then for the female and the guy as well, definitely that homocysteine. The homocysteine would be for sure, just to sort of find out if there is a bit of a methylation issue going on. And if there is, really high homocysteine, for the male, you've got your female on the prenatal, you definitely want to be putting him on a B complex as well. You want him on the Bs to help with that homocysteine. And then I didn't put it in there, but if you're asking my top ones, I do also think about vitamin D. Vitamin D is really important for fertility, immune health. So I'd probably put that one in there. And then I don't know if you do oxidative stress testing, but I do like that as well. So we can do a blood test here for oxidative stress or total antioxidant capacity. They'd probably be the sort of minimum. And of course, you can do so much more. I like genetics, but I know that is, I guess that's my specialist area. Not everyone's going to be doing genetics.
Yeah. Great. Well, thank you. I think we could probably go another hour.
I know. And don't leave me here too long. So I'm like, I didn't say iron. If there's anemia, this or it's that. But yeah, no, I just semen analysis, homocysteine, and maybe a D. You could do a lot with those things. Yeah.
Yeah. Okay, well, I guess we'll end here. I know it's later in the evening for you there. So thank you again, Denise, for joining us and just, giving us so much to work with. I know fertility isn't necessarily, maybe like a common thing that all the practitioners are working around, but this is definitely something that we can use. Just even looking at just general health, improving general health and well-being, looking at chronic diseases. So yes, thank
And
you.
I think if it's not, maybe you need to promote that you do have the ability to support natural conception, or even what I do is even supporting some of your clients through IVF. Of course, what we aim for is natural conception, but the reality is someone may have certain issues where they do need to sort of go through IVF. You still want to give them the best opportunity to get healthy sperm, healthy egg, and be able to then, for the woman to be able to maintain that pregnancy. Because I did actually, I will go in a second. Obviously, I just love to talk, everyone. But I did start trying to find statistics on assisted reproductive technology and IVF in Hong Kong. And it wasn't clear, but it did look like it was a really big issue, and infertility was increasing. ART rates are going up, and a lot of people leaving pregnancy until later. So I do think you've got the issue. So if you are a practitioner who's interested, perhaps it's more of a, even, at Gyra and with the practitioners, letting people know somehow that you can offer some support, because a lot of those people in Hong Kong may not even know that you're able to, probably never heard of homocysteine or methylation, and just think about folic acid. Oh God, help them. Yeah.
Yeah, that's a really good point. That's a really good point. I mean, just to help them through even if they are going through IVF, helping through that journey, and then through the actual pregnancy. Right?
Yes, not forgetting that part as well. So.
Yeah.
All right, everyone. Thank you, and it was so good to see you, Denise. And let's pray for 2023 that I might see you.
Yes, we would love to have you in Hong Kong again.
Oh, I can't wait to be back. So fingers crossed for 2023.
Okay.
Have a great day, everyone.
Thank you. Thank you all. The slides will be sent out and recording will be sent out as well. Thank you. Bye. Bye. Bye.